I am delighted to introduce to you our next speaker, Professor Darrell Irvine. He is the Eugene Bell Associate Professor of Tissue Engineering at MIT. He is an investigator of the Howard Hughes Medical Institute and has been a member of our faculty here at MIT since 2002. In September 2004, he was named to Technology Review's TR100, the magazine's list of young innovators who will change technology. Professor Irvine's lab works at the interface of materials science and immunology, studying immune cell biology and synthesizing new materials for vaccines and immunotherapy. His work builds on the premise that immunotherapy, beyond providing temporary reprieve from disease, can actually be curative, using the power of the immune response and immunological memory. Professor Irvine is helping to build better vaccines against diseases such as malaria and cancer. His laboratory uses microparticles made of new biodegradable plastics and building blocks made of DNA to create focused, tumor-killing immunotherapies and targeted vaccines with minimal side effects. They use nanoparticles as vehicles to deliver proteins directly into immune cells. Please join me in welcoming Darrell Irvine. We will have time for questions at the end of his remarks. Thank
you. Okay, I talk loud. So.
Well, it's for the recording.
Okay, great. Thanks for the introduction. So it's a pleasure to be here and tell you a little bit about some of the research that we're doing in my lab. And I could start by saying that I think Bob's story about his move from chemical engineering to a position in medicine for his postdoc, you'd like to think that that kind of thing is more common now, 30 years later, but it's really not. And I can say that my own postdoc, I was trained as a material scientist in my PhD. I did my PhD here at MIT, and then looked for something very different that I could do in my postdoc, even different from what other engineers interested in biomedical problems were doing. And so I contacted an immunologist at Stanford University, Mark Davis, who was well known. He was one of the investigators that discovered the T cell receptor that explained how T cells function. And so I spoke with some people in his lab who said that well, you know, Mark has a quite diverse group. In Bob's comment about hiring odd people really resonates. Mark's got a very diverse group. You know, he's got T cell immunologists, B cell immunologists, cell biologists, biochemists. And I thought, material scientists, though, that's a little farther outside of that sphere. But Mark took the chance and let me come to his lab where I learned a lot of immunology, and got very interested in thinking about how you can use materials to drive the immune system, to treat disease, to make vaccines, or for cancer therapy. And a lot of the underlying principles grow out of things that Bob started here at MIT in terms of what ways we use materials to do this. But we are very focused on what you could do with the immune system. And the problem of bringing immunity to fight cancer is that the immune system has not evolved to operate against cancer. So the immune system works by recognizing that when you have an infection, if you have microbes like viruses or bacteria, they're composed of molecules that your body does not use. And so there's a molecular recognition that this is foreign material and we need to respond. And that leads immune cells to go to your lymphoid tissues like lymph nodes, activate T cells and B cells, and then these cells go and hunt down these foreign-looking cells. And of course, the problem is that tumors lack these signals of identification. We don't know that a tumor is not a self cell in many instances. Your immune system does not know that. But that doesn't mean that there's no hope for bringing immunity to bear. And in fact, there's increasing evidence that your immune system in some patients, certainly not all, but in some patients, the immune system is playing a role. And an example is shown here from a paper from Science from a couple of years ago, where it was shown that if you look, this is looking at patients with colorectal cancer. And as you may know, there are these staging criteria that oncologists use to stage the severity of disease, from stage 1, which is the mildest, earliest stages of cancer, to stage 4, which is advanced terminal disease. And they found that if they asked the question, where are the immune cells? Are immune cells infiltrating these tumors? That they could actually make a better prediction of which patients would survive versus the traditional staging criteria. So some patients that were diagnosed as stage 1 cancer actually had very short survival, and some patients that had advanced cancer survived quite well if their immune system was infiltrating the tumor. So that tells us that the immune system might be brought to bear in a positive way. And so there are several approaches clinically right now that people use to treat cancer using the immune system, where you're trying to do a treatment that will get your own immune system working against cancer. So one of these is to inject antibodies that recognize tumor cells. Another is to try and generate vaccines against cancer. And some of you may know about the Gardasil vaccine that was just recently approved, which is, in fact, the first cancer vaccine because it acts against HPV that causes cervical cancer. But note that this is a special case because it's a vaccine against a virus which happens to induce cancer. And then there are drugs that can non-specifically stimulate the immune system, sort of inducing inflammation to get anti-cancer activity going. And then finally, there's this approach called adoptive cell therapy, and I'm going to say some more about this. And I'm going to tell you just a little bit about two areas of work that we're doing on these bottom two, trying to use what we do to drive the immune response. So one of the areas that we've been working in is to look at the idea of using materials to focus a drug on a tumor site, with the idea that if we could get the immune system going at one solid tumor site, then the immune system might be able to disseminate, locate tumors wherever they are in the body and eliminate them. And so we've been looking at materials that we could inject around a solid tumor, or maybe at a site where the bulk of a tumor had been surgically resected by a surgeon, and have that material loaded with molecules that would then diffuse into the tumor and drive an immune response by activating cells that are already in the tumor and also calling in their neighbors. And the idea of this local treatment is really that if we could have this material carrying potent immunostimulatory factors, we'd have some advantages that we're giving the same drug systemically. So a lot of the drugs, protein drugs like interleukin-2 that turn on the immune system, have a limitation that they're very toxic and pleiotropic because they're so potent in activating the immune system. So one of the advantages you obtain if you're treating a tumor locally like this is that if you look at the amount of this drug, and I'm looking at a particular protein called IL-15 that turns on T cells, if you gave that drug intravenously, as we would normally give these treatments, there is this very sharp spike in the dose that's in your bloodstream over time. And if you instead give it locally at a tumor site, that initial very sharp peak is blunted. So the toxicity that would arise from this high concentration is reduced. And more importantly, if we look at how much of the drug is actually in the tumor, here's the amount of drug that's getting into the tumor if we give it systemically. And that's simply because it's diffusing all through the body and not locally accumulating where you want it. And we can get about 40-fold more of that drug coming into the tumor at the peak when we've locally injected it around a given tumor. And the effect that can have of this dose treatment is quite significant. So this is an experiment where we inject around a solid tumor twice, once on day 14 and once on day 21, indicated by these arrows, these gel materials that are going to release this IL-15 stimulant that drives T cell responses. And you see that the untreated tumor here is growing out as expected, whereas the tumor growth is completely halted with this local treatment. And that translates into a major difference in survival. So these untreated animals all die by about 35 days, where 60% of them are still surviving with just these two local injections. So this focusing of a therapy it can be quite potent. And just as Bob was saying that low amounts of a drug, that toxicity is all about dosage, the same goes for our therapeutics. If we can make a difference of tenfold in the dose of drug that's getting to the tumor, that can be quite dramatic. Another problem we're interested in is this question of how you get the immune system to start going after the tumor. So and this is seen in human patients and echoes that data that I showed you from that Science paper. So in some cases, the immune system finds its way into tumors. This is tissue sections from a tumor, and the green fluorescent cells here are T cells that have come in out of these blood vessels in the tumor. And this is a tumor that's getting eliminated, whereas here's a tumor that's growing rampantly. And you see that one of the main differences is the complete lack of these T cells coming in. So how can we get immune cells to start coming into a tumor and knowing they need to be there? Well, one of the ways the immune system normally finds its way when there's an infection is that the host cells that recognize there's an infection going on will secrete factors called chemoattractants. So they're secreting proteins, and as those proteins diffuse away from the secreting cell, cells like T cells and B cells and other immune cells can sense that there is this attractant coming from this cell. They basically detect that the concentration of this molecule is getting greater as they move this direction. And so they undergo a process called chemotaxis, where the cells actually move to that area of the body where this signal is coming from. And so we started thinking about, well, we could use these controlled release materials like these microspheres. Instead of loading them with a drug to treat a therapeutic drug, what if we loaded them with an attractant that would try to synthetically mimic this process of drawing cells to a site? And hopefully this video will work. So we've been developing these materials with the idea that let's inject a particle into a tumor that's going to call immune cells into that tumor. And so the way we model this in vitro to see if we can, to test this idea, is we have a microsphere here and a microsphere here, and they're suspended within a collagen matrix. So it's a mimic of tissue, and we have lymphocytes all around here. And this is one kind of immune cell called a dendritic cell, and over here is a T cell. And with any luck, this is going to play. Yes, all right. So what you'll see here, and this will loop, you'll see these dendritic cells come in, and we're imaging over many hours here. So this is sped up time lapse. And what you're seeing is that this particle that's releasing this attractant really dramatically, all of the cells in the nearby environment are drawn right to that site. And we can do the same thing with T cells. So you see these T cells that were at the first frames of the movie come swarming in, and then they stay sort of buzzing like bees around this bead. And this is exactly the kind of thing that we'd like to be driving in the tumor environment. Call these immune cells from the local blood cells into the depths of the tumor where they can start recognizing tumor antigen and really trying to destroy it. So that's one area that we're working on. Now the second part of my talk is really going to get into the nanotechnology side of things that we do. And here, what we're trying to leverage is the fact that your T cells have the impressive capability that if they're in the right state, they can see a tumor cell and they can kill it. So if we could get T cells going, they could be a really potent cancer therapy. And there's an approach in clinical trials, both at the National Cancer Institute and at a few cancer centers around the country, called adoptive cell therapy, that is basically trying to take this idea to the limit and say, let's make an army of T cells from each patient that would be able to attack and destroy a tumor. And the way it works is like this. You take, for example, you could take a tumor biopsy from the patient, and you get the T cells out of that tumor biopsy, and there are ways to identify which T cells are killer T cells that will recognize tumor. And the idea is, well, let's grow those up in cell culture so that we have tons and tons of these cells, and then inject them back into the patient with hopes that they would go anywhere in the body, localize, and destroy the tumor. And this, as I said, this is in clinical trials right now. It's been tried in a few infectious diseases, and in particular in melanoma, it's been tested a lot. And it's a great idea, and the two limitations right now are basically that it's hard to find these T cells, so we get very few of them out. so that it takes a long time to isolate them. And in some cancers, it's very difficult to find any T cells that are tumor specific. And then the second problem is that when we inject them back into the patient, they will often quickly die out. And this is just a limitation on our understanding of the biology yet, that we don't know how to make these cells stay functional. Well, other people have essentially come up with a good idea to solve the first problem of not having enough of these cells. So they've said, let's use genetic engineering, and basically I think this cartoon captures all that you need to know. Let's put a gene into the patient's T cells that makes them have a T cell receptor that will let them recognize and kill tumor. And this has been done in human patients, and it's safe. And it basically means that I could take T cells from any person and make them anti-tumor T cells, which is very exciting. So that piece of it seems to be a problem that we can solve. And a few years ago now, this was demonstrated in human patients. And you could see amazing results, such as this is a patient with metastatic melanoma in the liver, and you see this large liver metastasis that's regressed over a period of months by this therapy. But there is still this second problem that if we really want to cure people with this approach, we have to make these cells stay functional longer. And as I said, one of the problems with these immunostimulatory drugs, we do know some drugs that could do this, but they have very serious side effects. So we can't just inject tons of IL-2, IL-15, and so on into patients. So we've started looking at an idea. So the genetic engineering that they're doing right now is sort of engineering the cell from the inside out. What if we started engineering the surface of the cell with some synthetic nanoparticles? And here's the idea. We're going to attach onto the surface of the cell a drug-loaded nanoparticle, much like the kinds of materials that Bob was talking about. We're going to load a drug into that particle that's going to get carried around with this cell wherever it goes. And this is kind of a flip side of the idea that Bob was talking about. Instead of trying to use nanoparticles to get a tumor-killing drug to the tumor, here what we're going to try and do is get a drug that acts on the carrier cell and makes that cell work better along its normal line of function. And the idea is that we'd like to be able to inject these T cells, and they will go to the tumor just as they always did, except now they're carrying this little package of drug that as the drug comes out, it binds back onto that T cell and keeps it going, keeps it alive, keeps it functional. And I won't get into the details of the chemistry here, but basically it's very simple. It turns out that T cells have a surface chemical group, thiols, on their surface, and we can just do very simple reactions that link this drug-carrying particle onto the cell surface, and they don't mind. It's non-toxic. And what's amazing is that you can do this process, link any number of particles onto the cell. So these are T cells here in green, and the purple down below is showing the particles that were attached on these cells, and an overlay is shown up here. And amazingly, these particles will stay on the cell surface for a week at a time, so that any drug that we load in here could be continuously released and act back on that cell. And I think I'll skip some of these more minor details. But the amazing thing is that even if we stimulate these T cells to proliferate, which they need to do when they're going after the tumor, the particles stay on the cell surface. So a process called endocytosis, where cells would normally be taking things in off their surface as part of their normal homeostasis, does not happen for these particles. And that's important because it means that the drug that we're releasing here can keep acting on the cell for that entire time period as time goes on. Okay, and another side effect that's a really cool sort of benefit we get out of this chemistry we're using is that the cells don't mind having the particles on their surface. They can, for example, migrate through collagen tissue matrices like I'm showing here. So these are T cells crawling through tissue. And this is overlaying in red is the fluorescence from particles on the cell. And maybe you can see some of these cells go running through the field. When T cells are migrating, they take on this hand mirror shape. And this is the front of the cell. He's going that way. and this is the tail called the uropod. And amazingly, many of the proteins on the cell surface get moved to the tail when they're migrating to keep those receptors out of the way. And our particles, because they're piggybacking onto these proteins on the cell surface, they also get pulled to the tail. So an unexpected benefit of this chemistry we're using is that as the T cell's running around, the particles are sort of out of the way. They're sort of being carried at the back end and hopefully don't interfere with what the T cell would normally want to be doing in the tissue. Okay, so, but the real test is, can a T cell that's zooming through the bloodstream squeeze through an endothelium and enter the tissue normally when we've loaded these nanoparticle drug delivery vectors onto their surface? Because that's the real test here. So I'll show you an example. One of the ways that we model this using mouse models of cancer. In this model, we induce tumors in mice that we can image with a technique called bioluminescence imaging. So we have this false color scale here telling us where the tumor is. And in fact, we're doing an experiment where one tumor on this side is recognized by our T cells, and the other tumor is just a control tumor that the T cells will not recognize. And then we're going to treat these animals by either injecting nanoparticles alone, T cells alone, or T cells that have the particles stuck on their surface. And we're going to ask, can the T cells get the particles into the tumor? And first, if we image where the T cells have gone, you see that if you compare this case, this is the T cells alone, and as expected, they go to the tumor that they recognize. And if we look at the T cells carrying particles, they also go to the tumor, and qualitatively and quantitatively, they're getting there just as well. So the particles didn't block T cells from moving in vivo. And what's really amazing is that for these particular particles we're using, when they're injected intravenously, mimicking how we would normally give drugs to a patient, they accumulate very poorly in the tumor. You can't see any fluorescence in this imaging approach at all. But the T cells that have carried particles, carrying them physically with them into the tissue, you see this strong signal that's telling us they've brought a large number of particles into the tissue. And that's quantified over here, where basically if we're measuring by the fluorescence signal how many nanoparticles get into the tumor, it's barely above background what we get to the tumor with the blood injection, but the T cells carry this massive dose in. And so this is really exciting because it says that the lymphocytes can home these drug carriers along with them into sites where the particles would normally never get to. So then the next question is, can we do anything therapeutically useful with this approach? Because that experiment was with empty drug carriers. And so, as I said, we've sort of started by focusing on the opposite sort of problem from what Bob showed you with his anti-cancer drug delivery. We're delivering two drugs called IL-15 and IL-21 that are in clinical trials in human patients. We're putting both of these drugs together into these particles. They're going to slowly be released, and the idea is we want them to bind back to the carrier T cell. And these drugs have been implicated in driving very potent anti-tumor T cell activity. So they keep the T cell alive, they help it proliferate so you make more of them, and it keeps that T cell killing tumor when it would normally lose its function over time. And the drug and the materials that we're using will release these drugs over about a week once they're attached to the cell. So we'd expect our drug to keep going for a week. And this is the therapy we're gonna test here. So we have animals that have a model of lung metastasis. So we're doing melanoma tumors that are metastasized to the lungs and bone marrow, so mimicking advanced cancer that we can image again with bioluminescence, so we know where the tumors are. And we're going to treat these animals. We're either going to mimic this adoptive cell therapy that I said is being tested in humans already, giving the animals T cells that will recognize this tumor, or we'll give T cells plus an injection of these two proteins, IL-15 and IL-21, to try and keep those T cells going. But we're going to give one single injection because we want to mimic the limitation that you can't keep injecting these without toxicity in people. Or we're going to take the same dose of drug and we're going to attach it to the cells directly, loaded in these particles that will slowly release the two drugs out over time. And basically we want to know, is this approach going to be as effective as the systemic drug, and how will these three cases compare? So the first thing we do is we image where the T cells are. So here we're looking over time at day 0, day 4, day 8, day 12, and so on. And we're using this bioluminescence whole animal imaging to say, where are the T cells once we inject them into these animals? And this is just day 0, again, where the tumors are when we start the treatment. If we give the animals just the T cells, then you see that they go where the tumor is, but then they quickly die out. And on that false color scale, you see that there are not that many. They're in the cold side of the spectrum, saying there aren't that many T cells surviving very long. And if we give them this injection of the IV drug protein, they actually don't do much better. There's a little more signal here, but they also die out by about day 6. What we were really amazed to see is that if we give them instead this approach where they're carrying the drug with them, where every cell has its own sort of support kit, about 2 days after treatment, the T cells start proliferating like mad. They keep proliferating for about 6 days, and then they contract back down, and they become a stable memory population in the animal. And that's quantified over here. So the T cells carrying drug, there's about 100-fold more of those T cells at the peak compared to the control treatments. And the effect on survival is also dramatic. So the untreated animals, the tumors grow out, and these animals need to be sacrificed at this time. The animals that got T cells alone, or T cells with just one injection of the drug, they transiently clear the tumor out, but then the T cells die out and they didn't kill all of the tumor, so the tumor starts growing back, exactly what happens in patients. And by day 16, the tumor's already growing back, and you see that if you look at their survival, they survive a few more days and then they all succumb. But the T cells that were carrying their own drug have completely eliminated the tumor by 2 weeks. And if we follow them out for 100 days, they're essentially cured. So in this model, this boost to the adoptive T cell therapy can be curative. And we're hopeful that this might be useful in other settings besides just the case of directly killing cancer. And I'll just quickly finish up by showing you one more example. So another common procedure during cancer therapy is when patients are on high-dose chemotherapy and radiotherapy regimens, they may get a bone marrow transplant, which is done clinically every day across the river at Dana-Farber Cancer Institute. One of the limitations of getting a stem cell transplant is that during the time, because the stem cells are the source of your immune system, and your immune system has essentially been ablated by your chemotherapy treatment, there's going to be a window of time, which can be as much as 6 months, when patients are greatly immunosuppressed. And if they get a cold, it can be fatal at that stage. And so there's a lot of interest in ways to make bone marrow transplants repopulate a person more quickly. Basically, you need these stem cells to divide and repopulate your immune system as quickly as possible to recover from this treatment. So we can do the same sort of approach. We're going to load drug-carrying particles onto stem cells, give them into an irradiated animal to mimic a bone marrow transplant. And in this case, we're going to give them a small molecule drug that, again, will release over from the particles for about a week. And this drug has been shown, if you give it every day to animals, it enhances the kinetics of bone marrow reconstitution. And so here's this experiment. Again, we're using this whole animal imaging approach to look at where the bone marrow reconstitution is happening. So you inject stem cells that are bioluminescent, and as they repopulate the animal, eventually all of the tissues in the animal become bioluminescent because you're regenerating luminescent T cells and B cells, erythroid cells, et cetera, all of the cells that the stem cells become. And you see that this takes about 3 weeks, and you've got a nice strong signal there in the control animals. If we give this drug, TWS119, that's been looked at as a way to enhance the therapy, but we give a very tiny dose, again trying to minimize side effects, then at that tiny dose of 1.5 nanograms, there's essentially no difference between the control and the animals that got the drug. They reconstitute, but it takes several weeks. If instead we give them stem cells that were decorated with these particles loaded with that same dose of drug, then even a week out, we can start to see the stem cells have started to expand, and at 2 weeks, they've already expanded more than the animals over here in the controls that were out at 3 weeks. So we're really speeding things up with this simple approach. And this is just measuring that another way. This is saying how many stem cells have repopulated in the animal at 2 weeks out, and with the cells carrying the particles on their surfaces, they're about 5-fold more reconstituted at that time point. So, and at the end of the day, they still make the same B cells, T cells, myeloid cells, erythroid cells that the stem cells would normally make. So we don't change the eventual outcome. The stem cells still work the same. We just make it happen more quickly. Okay. So I think I'll wind up with that, and I hope I've tried to illustrate with these couple of vignettes some of the ideas we're looking at. We're really excited about this application of using nanoparticles as a way to engineer cells directly. And though one of the limitations of adoptive T-cell therapy is that it's a very laborious clinical approach, because you have to isolate these cells from patients and have essentially patient-specific medicine. With this approach to enhance that therapy, it may be one of the most promising approaches for advanced cancer, and we're quite excited about that. And in this second little bit, I told you about how this could be applied to stem cell transplants, and here we're able to reconstitute animals about 5-fold faster with a drug that's already in patients for diabetes and some other conditions. So hopefully perhaps a low-hanging fruit to get into the clinic. So with that, I'll finish, and I'll just note some of the great students. So a great postdoc in the lab, Matthias Stefan, has worked on this particles on cells work I told you about, and the injectable gels and chemotaxis work I told you about was done by grad student Brandon Kwong in the lab. So thanks, and I'd be glad to answer any questions.
Models there, do you see any negative consequences to the sort of massive overproliferation that you're seeing, that transient period?
Yeah, so we were certainly nervous when we were running that experiment and saw that proliferation happening in the middle of the experiment. But in fact, we've followed those animals out now more than half a year, and there's no sign of, one of the worries you would have would be potentially the induction of leukemia, so your T cells themselves become a cancer. We think, though, that the reason that didn't happen is that we're using the normal immune homeostasis mechanisms. We're just making them work a little bit more efficiently. And in some infectious diseases, you can see this level of T cell proliferation. It's actually in cancer, it's blunted, so you don't see it. So this is something we definitely are very carefully looking at. But so far, it looks to be safe, and the cells contract back down to a population that sticks around, which would be a great thing in case the cancer comes back, but they're not out of their normal homeostatic control. Yep.
We saw the mouse model completely clearly. Did you check for dormant cancer cells that would re-energize, which is very common in melanoma?
Yeah, so we're trying to set up models to look at that more carefully. Yes, so the question was, could there be dormant tumor cells that reside somewhere that were not killed that could come back later? We can't detect them in that system where we're using bioluminescence to detect them, and we've looked in some of the major organs and don't find them. But we need to set up other models to look carefully for that. And so that We've seen that we don't see tumor growing back out at half a year, but we're trying to set up models now where you can more carefully look for those cells because they're hard to detect.
Yeah, those one slides that you showed where you had the nanoparticles of the IO with the 21, I think it was, and the animals went completely red in terms of the animal.
Yeah.
Is there a lot of this inflammation associated with that, or it looked like the animal was going through a pretty big period of stress there?
Yeah, so well, and I should say that's a little bit of an artifact of the way we're measuring them. So the gain is set to the same gain we image the control animals so that we can see that there are still T cells in the control animals. But so there's a lot of proliferation going on, but it's not as though the animals are outside the window of what their immune system can normally be doing, as I was saying, during a serious infection. So this is the kind of thing that in people is going to look like flu-like symptoms, but
So there wasn't any observable stress in the animal?
Right.
Even though the image went completely red?
yeah, that's right. That's right.
Darrell, can I ask you to comment on the translation of these observations? I actually mentioned your work in my introduction yesterday as an example of things moving rapidly in the laboratory that have clear and relatively short-term implications for the clinic. So with the adoptive cell transfer with supercharged T cells decorated with stimulatory nanoparticles,
Yeah.
can you comment on that specifically, both with respect probably to the NCI, but also local interactions with the Dana-Farber and others?
Yeah, so one of the reasons that we went down this path in the first place is that there are several clinical centers, the NCI, the Fred Hutchinson, Sloan Kettering in New York, that are doing this therapy with T cells in patients right now, even this genetically engineered T cell work in patients. So what we hope is that this is one more step that where you just mix the T cells with the particles, they link on, and then they go into the patient. So a minimal perturbation to what they're already doing. And so we're basically looking now to get contacts with some of these clinical centers. So in Boston, T cell therapy isn't being done right now. However, as I mentioned, bone marrow transplants are done in huge numbers here in Boston. So for the HSC transplants, we are talking to some people at Dana-Farber Cancer Center across the river about the prospects of starting to think about early stage trials. And as Bob said, one of the hurdles is you really have to bring a company in at that stage because academic labs are not really suited to make FDA quality materials to put in people and to make lots of it and so forth. But this is also something that MIT is pretty good at. So. Okay, thank you.
Very much.